Welcome to Module 4 of Working with EEG Data. Our first section will basically be all about introducing EEG. And here is what's going to be covered in this section. So for this lesson, we'll start on some EEG introduction by talking about the neural basis of EEG. And then for the other upcoming videos, we'll cover brainwaves, some EEG terminology, and the common types of EEG data analysis. So here's what you guys should already know so far. Neurons are cells in the nervous system that communicate using both electrical and chemical signals, and action potentials are electrical signals that travels down the axon and are used to send information to other neurons. Postsynaptic potentials are different from action potentials in that they are graded potentials, meaning they are changes in the membrane potential that vary in size. In other words, postsynaptic potentials are not all or none signals, unlike action potentials. This distinction would be important for understanding what the EEG detects, and which is what we'll cover in this video. So the key questions that we're going to cover include: what is an EEG and what can it detect? What are some properties of EEG signals that allow us to detect brain activity? And what are some advantages and disadvantages of EEG? So as a reminder, EEG is placed on a person's scalp to detect electrical activity of neurons in the cerebral cortex. It can be used to measure brain activity that occurs during an event or to measure spontaneous brain activity. It detects the signals created when populations of neurons fire at the same time. And EEG is primarily just measures postsynaptic potentials, or changes in membrane potential that are elicited by neurotransmitters binding to receptors on the postsynaptic membrane. Action potentials in the axons do not contribute much to the ongoing EEG activity, as they are too short. They go in too many directions relative to the surface of the cortex, and they are not synchronized. The roots of the EEG lie in the work done by English physiologist Richard Keaton in 1875. He made electrical recordings from the surface of dog and rabbit brains using a primitive device that is sensitive to voltage. The human EEG was first described by Austrian psychiatrist Hans Berger in 1929, who observed that waking and sleeping EEGs are distinctly different. Today, the EEG is used primarily to help diagnose certain neurological conditions, especially seizures, epilepsy, and for research purposes. Notably to study the stages of sleep and cognitive processes during wakefulness. So the physiological basis behind an EEG signal can be explained by considering the electrical activity of a single cell activated by an afferent pathway. So the incoming signal in the synapse gives rise to a postsynaptic potential resulting from the positive charge ions rushing into the cell. So as this current propagates down the neuron, the size of the EPSP, or excitatory postsynaptic potential, decreases, which leaves a relatively negative charge in the a negative charge here in the extracellular space immediately surrounding the synapse. A dipole is therefore created with separation of charge vertically oriented in the cortex. So dipoles are a pair of equal and oppositely charged or magnetized poles separated by a distance that can be identified by the electrodes placed over the scalp. Signals cannot be detected from single neurons because the potentials are far from the scalp and have a very small magnitude. So the signal detected is the summation of all the dipoles created at each of the 1,000 neurons. And in order for the brain activity to be easily detected, the signals that are summed must have two main properties. So the first property is that the neurons must behave similarly. In other words, a group of neurons must be activated all together so that the same charges are propagated at the same time. The second property is that they must be oriented in the same direction. So if these two properties aren't met, the charges cancel each other out, and so the electrical activity isn't detected. The pyramidal neuron in the cortex, which is shown here, is a great example for satisfying these properties. All right. So there are several reasons why high temporal resolution techniques such as EEG are exceptional tools for studying neurocognitive processes. The first reason is that these methods capture cognitive dynamics in the time frame in which cognition occurs. So cognitive, perceptual, linguistic, emotional, and motor processes are very fast. Most cognitive processes occur within tens to hundreds of milliseconds. Furthermore, cognitive events occur in a temporal sequence that may span hundreds of milliseconds to a few seconds. So high temporal resolution techniques are well suited to capture these fast dynamic and temporally sequenced cognitive events. And by comparison, the temporal precision of the hemodynamic response obtained in fMRI, for example, is around 2 to 3 orders of magnitude lower than that of the electrophysiological response. So this is why most brain-computer interfaces, including prosthetics and many other neurotechnologies out there, take advantage of EEG's high temporal resolution. And a second reason why tools such as EEG are advantageous for studying neurocognitive processes is that they directly measure neural activity. The voltage fluctuations that are measured by EEG are direct reflections of biophysical phenomena at the level of populations of neurons. And furthermore, oscillations that can be observed in the EEG signal are direct reflections of neural oscillations in the cortex. So this can be seen as an advantage over currently used MRI-based functional measures, which do not measure neural events directly and have a more complex relationship between what they actually measure and what can be inferred in terms of the kinds of neural dynamics that produce or correlate with a hemodynamic response. And a third advantage of EEG is that the EEG signal is multidimensional. Although you might conceptualize EEG data as two-dimensional or basically voltage changes over time and space, where space is measured through different electrodes. In fact, EEG data consists of at least 4 dimensions, which we'll talk more in detail later. It's time, space, frequency, and power and phase. So we'll discuss what they are later on in this section, but for now, you should know that this multidimensionality provides many possibilities for specifying and testing hypotheses that are rooted both in neurophysiology and in psychology. So the brain can be conceptualized as an enormously complex biological system that uses a multidimensional space for information processing, representation, and transfer. Of course, EEG is not perfect. EEG is a powerful and insightful brain imaging technique, but it is not well suited for addressing all research questions or creating types of neurotechnology that require high spatial resolution. EEG, due to its poor spatial resolution, is not well suited for scenarios in which precise functional localization is important. If your research problem involves testing whether different striatal subregions are preferentially involved in 2 different cognitive processes, EEG will be of little use to your research. However, if your research problem requires dissociating activity in parietal cortex from activity in the frontal cortex, the spatial accuracy of EEG is likely to be sufficient to confirm functional or anatomical dissociations at this scale. Nevertheless, EEG is likely to be a suboptimal method for any research questions involving where in the brain does process X occur or is information Y stored. Another EEG disadvantage is that EEG is not well suited for testing hypotheses about deep brain structures. Although activity from deep brain structures can be measured with EEG in some cases, which we won't really have time to cover, but EEG may also be another reason that it could be a suboptimal brain imaging technique for questions concerning cognitive processes that are slow and that have an uncertain and variable time course. So that sort of defeats the purpose of using EEG because we mostly want EEG for its high temporal resolution. The extremely high temporal precision of EEG might be disadvantageous in some cases or some other research questions. But many EEG analyses become unreliable when the cross-trial jitters in timing are longer than a few tens or hundreds of milliseconds. The relatively low temporal position of fMRI is better suited to studying slow cognitive processes. So some social and emotional processes might have slow and uncertain time courses that can be more difficult to interpret with EEG. So To summarize, EEG can be used to measure brain activity that occurs during an event or to measure spontaneous brain activity. In order for brain activity to be easily detected, a group of neurons must be activated altogether and they must be oriented in the same direction. The advantages of EEG are high temporal resolution, direct measurement of neural activity, and multidimensionality. The disadvantages of EEG are poor spatial resolution and inability to measure deep brain activity. And for the next video, we'll be talking about brainwaves.